So good afternoon. Thank you for having me today. I will briefly introduce the background. Okay, let me choose minimize. So memory deteriorates with ages, and this threatens independence and well-being. However, our ability to support memory remains limited. I think part of the reason is that we do not yet know how the brain adjusts its internal state to encode new informations. We all experience this. Sometimes information is easy to remember, but in other times it just doesn't stick. So one candidate mechanism would be related to Saccade. Let's do an interactive experiment to show why I had this idea. So please take out your phone and open the camera app. Yes, we are doing this. Make sure it's in video mode. And then switch it to the front-facing camera like we are doing a selfie. Hold it close enough that your face fills most of the screen. And let's start video recording. Now try to answer this question. What do you have for dinner last night? You can stop the video recording. And when you play it back, if you watch your eyes closely, you probably see them darting. Here is an example of me when I was asked the same question. So this observation suggests that saccades are intriguingly tied to memory. But what is the neural mechanism coordinating saccades and memory processes?
I'm sure it wasn't because you had your hand in an electrical outlet.
That's not part of the reason. For the rest of the talk, I will refer Saccade-related neurodynamics as changes in neural activities that have temporal relationship with the time, with saccade timings. Here is an example. This is the neuron from superior corticus from a rodent model, and the x-axis represents time relative to the probe presentation, and the magenta line represents the saccade timing. We see the increase in firing rates, which is one type of saccade-related neurodynamics. People started to study saccade-related neurodynamics mostly in order to understand how the brain supports visual perception. So when we see, when we move our eyes, the external world sweep in, and the brain needs to address these challenges. One is to suppress the intrasaccadic motion sensation that arises because the eye movement. Second is that the brain needs to keep in register where in external world the pre and post saccade images originated from. And the saccade-related neurodynamic is believed to contain a copy of the motor signal, aka an efferent copies. And the identified pathway is mainly from superior corticus to frontal eye field. And in recent study, including ours, we started to show saccadic modulation is more widespread than previously recognized. Here, the color represents the proportion of electrical contacts that show consistent waveform changes after saccade. I will now describe how you extract those saccade-related neurodynamics in humans. Here is our experimental setup. We record both local field potentials and eye tracking data from invasively monitored epilepsy patients. In these specific settings, the patient is freely watching images displayed on the screen. And below are the two traces. One represents the gaze coordinates and the other represents local field potential recorded from left and right temporal lobe. So x-axis represents time relative to image onset, and zero, before zero, the participant was watching a fixation course. So at that period, the local field potential seemed uncorrelated, But after the subject make eye movement, their waveform began to show similarity. So if the waveform were consistent after Saccade, we would expected the intertrial correlation different from baseline. Here's what we observe. So we pick an example of a contact located in the middle temporal lobe. And the right plot shows the intertrial correlation. In our data, we actually observe a bimodal distribution, meaning in half of the trial pairs, the waveform were positive correlated, and in the other half, they were negative correlated, which suggests the presence of two consistent waveforms of opposing polarities. When we use hierarchical clustering to identify them, here's what they look like in terms of waveforms. I'm not going to go into the full pipeline here, but the detailed method will be included in our preprint. The key point is when we are able to use a metric to quantify the consistency of waveform based on the intertrial distribution. And here is what we observe. The Saccade-related evoked potential widespread, especially in the temporal and parietal lobe. And the inset here shows the electron location across all the subjects. Some of you may ask, could these Saccade-related evoked potentials simply be the ENG signals originate from the extraocular muscles? If that was the case, we would expect to see the evoked potential P amplitude to increase with larger eye movements. So in our studies, we have both electroauriculagram, EOGs, and intracranial EEG. The EOGs mainly captures muscle activity. We see what we would expect, that there is a linear relationship between eccentricity, the angular distance of the eye movement, and the peak amplitude of the evoked potentials. However, in our intracranial signals, we do not find such relationship. In fact, the eccentricity only explains less than 1% of the evoked potential peak amplitude, and the right part is a summary of our results. So here I confirm the existence of Saccade-related evoked potentials in humans. The next question is, do these evoked potentials influence visual encoding outcomes? Because they are not just represented in the pathways, which we start to have motor signals, they are also present in the brain region that is associated with memory. When we first introduce the data structure, we collect data from 8 subjects. Each subject finish tasks, multiple tasks depending on their duration of their stays in the epilepsy monitoring units. And each task has multiple sections with a total about 180 trials. Each trial here represents the 3-second image presentation. There could be multiple saccades happening within a trial, and we report Saccade-related evoked potential across electron contexts during one saccade. And each Saccade-related evoked potential will labeled either remembered or forgotten, depending the testing result on the next day. Here we just focus on Saccade-related potential during the encoding phase. Here is an example of the reconstructed Saccade-related evoked potential during one saccade. The time trace is high dimensional. Therefore, learning the mapping between these Saccade-related evoked potential and memory outcome could be computationally costly and hard to interpret biologically. Therefore, we propose a model that parameterizes this waveform using 5 parameters. This includes the pre-saccade oscillatory component using 2 parameters, the power and phase, and half oscillation cycles before the evoked potential onset. And we use 3 parameters, latency, peak time, and the amplitude to characterize the evoked potential. With that, now we want to learn the mapping between these parameters and memory outcome. The model we were using is random forest model because it's capable to learn complex relationship through a set of decision rules. I remind once again, the inputs are the 5 Saccade-Related neurodynamic parameters, and we were using 5-fold cross-validation for training and evaluating the models. The evaluation matrix is balance accuracy because it accounts for the imbalance in the sample numbers between classes. And the conclusion is, yes, those parameters predict visual encoding outcomes. When we look at each individual electrical context, the observed balance accuracy is about 56%. In these plots, the distribution to the left represents the new distribution generated by shuffling the labels of the data and retrain and re-evaluating models. We can, of course, use all the information by averaging the predicted probability across electrical contexts during one saccade. With that, we see a slightly higher balance accuracy at about 63%, which again exceeds all values in the new distribution. So we now confirm that Saccade-Related evoked potential play a role in visual encodings. Now we want to explain the learning mappings in the random forest model. To do that, I use Shapley additive explanation framework, which basically quantify a marginal contribution of a feature value to the final predicted probability for each sample. Here's the equation. An intuitive understanding would be the Fs of xi is the expected predicted probability with knowing feature set S. So here we present the Shapley value for feature j and sample I would be the subtraction between two expected predicted probability. In the case of a random forest, is much simpler. Each leaf is associated with the predicted probability, and the expected predicted probability is the multiplication of the leaf value with the probability of a sample reaching that leaf. So here's an illustration. Again, I take one context, I plot their Shapley values. So x-axis represents the Shapley value. The y-axis is the 5 parameters we were using for each context, and the color here denotes the feature value. Take latency, for example, we see higher Shapley value for lower latency value. What does it tell us? It tells us an early evoked potential is associated with successful encoding. But again, interpreting the Shapley value is again very high dimensional, and the human like to think with simple decision rules. So in this case, I actually reduce the Shapley value into simple loop, including threshold and the direction. So here I illustrate how I did this. I first order the Shapley value by the feature value and find a change point for the feature value that can differentiate their average Shapley value. So here we have a threshold and the directions. Based on this simple decision rule, when the feature exceeding a value is associated with successful encoding, we were able to have a slightly higher balance accuracy than the full model. This time is about 76%. So these results suggest that we were able to extract the meaningful mapping from the random forest model. Let me quickly showcase how we can use these mappings to gain neuroscientific understandings. Here I will take the amplitude of the Saccade-Related evoked potential as an example. I use 2 color bars to represent direction. So there is a red color bar and blue color bar. When a dot are you going to show brain with colors. So if a color is in the red color bar, okay, it means that the amplitude, when the amplitude is higher than the threshold, is associated with successful encoding. Here's how it looks like. So we found in general, higher evoked potential amplitude is associated with successful encoding, and this is particularly true across the frontal, parietal cortices and thalamus. So to sum up, I show Saccade-Related evoked potential in humans. I demonstrate that they are not ocular muscular artifacts, and I show that they predict visual encoding outcome. With that, I want to thank my supervisors for their support and guidance, Dr. Erin Luther, Dr. Peter Brunner, and Dr. John Mealy, and our collaborators for their insightful discussions. Moving forward, I think it will be very exciting to navigate both the neurobiophysics of neuromodulation methods and computation models to try to modulate our memory outcomes based on the Saccade-Related evoked potentials. I'm happy to take any questions.
So we have plenty of time for questions.
Please.
The premax that you were showing of this related modulation, what is kind of odd that but like V1 had very different modulation when each of the students, whereas the rest of them are pretty symmetrical. Do you have any idea why that would be?
Yes, I will repeat the question. The question is, in these spatial maps, the V1 is different than the rest of the brain, and why would that be? Here I apologize for the color scheme. The right V1 is color coded by black, means we have no electrical coverage on that region. That's why it's black. Yeah.
Sure, there's negative encoding correlates of are they related to the direction of saccade anymore?
Nope. So we actually did very careful to eliminate that possibility. What we did is we have two classes of waveforms, and each is associated with a certain number of saccades. We look at the circular histogram for the saccade direction for the two classes, positive polarity and negative polarity, and their relay statistic doesn't show a directional peak. Therefore, we don't think they are related to saccade directions. So I need to repeat the question. The question is, we have two classes of waveforms. Are they related to the saccade directions? Thank you.